All right, welcome back. Yesterday we started talking about the immune system, Chapter 31, and we learned about the two different types of immune systems. You have innate immunity and adaptive immunity. We described white blood cells, and then we went over the four major components of the immune system. And then we talked about what would happen if a bacteria landed on the skin, and then what would happen if the bacteria landed inside the nose. And then we left off, or we finished talking about the second line of defense. So we should be on your notes at inflammation. And inflammation is the response when tissue is damaged because of infection or injury. When you have inflammation, you get redness in that area, swelling, and you feel this heat near the damaged site. Inflammation can be painful, and it's a very important response, though. What it does is it isolates the area to stop the spread, so that damage will not spread any farther than that inflammation location. It recruits cells to the location to kill any pathogens, and it promotes healing. Response to inflammation, the cells that do this are called mast cells, and what they do is they adhere to the skin and the linings of the organs, and they release all these chemical signals. There are three major chemical signals that mast cells release. First one is tumor necrosis factor. This is a cytokine protein that will kill target cells and activate immune cells to do their job. Second type of chemical signal are prostaglandins. This is a fatty acid derivative, and that will initiate the inflammation in nearby tissues. And then the third chemical signal is histamine. This is an amino acid derivative that increases the permeability of blood vessels to white blood cells so that they can act in nearby tissue. All right, so suppose you had a splinter. And you can see in the diagram the splinter is going right through the skin. And what's going to happen is that this damaged tissue is going to attract those mast cells. Right away, it's going to release histamine. Okay, you can see that in the picture. The mast cells are releasing that histamine, those little purple dots, and that histamine is going to diffuse into blood vessels. Also, the first chemical signal on our list, tumor necrosis factor, is also going to be released that will diffuse to phagocytes. All right, so what happens next? Histamine that was released is going to cause the vessels to dilate, and so the blood vessels become leaky. And then complement proteins are going to leave the blood vessels because they're leaky, and they will attract and activate phagocytes to that area. Tumor necrosis factor is going to be released, that signal, and that's going to stimulate phagocytosis. So we're going to get the pathogens there. The blood plasma and the phagocytes will move into the infected tissue from the blood vessels. Okay, what happens next then is that phagocytosis is going to engulf bacteria and the dead cells. Histamine and complement signaling will stop because they don't need it to be there anymore. Phagocytes are no longer going to be attracted once that signaling stops. And then a growth factor from platelets that are there will stimulate mitosis, epithelial cell division, and that will eventually heal that wound. All right, so after inflammation, pus will follow. Pus is going to form. Pus is a mixture of leaked fluid and dead cells. In other words, it's bacteria, white blood cells, and damaged body cells. Pus is a normal result of the body fighting a disease or some type of inflammation. Eventually, the pus will cease and will go away, and that pus gradually will be removed by macrophages. Inflammation is a good thing, but however, it can cause medical problems. An allergic reaction can occur. There are non-self molecules that are normally harmless, but what those things can do is they can bind to a mast cell, and that causes a release of histamine and subsequent inflammation, so more inflammation. along with the symptoms that you get when you have allergies: itchy, watery eyes, and possible rash. This non-self molecule that's doing this and causing this problem could come from food or something from the environment, like pollen, for example. Another problem that is caused by inflammation is an autoimmune disease, where the immune system will fail to distinguish between the self and the non-self items, and so the body ends up attacking tissues in its own organism's body, and that's obviously not good. And the third problem that could happen with inflammation is sepsis. The inflammation does not remain localized, and it extends throughout the body with the dilation of blood vessels. And so if this is coupled with blood pressure dropping, it could be a lethal situation. All right, so with inflammation, in most cases, innate immunity is good enough, but in some cases, when the pathogen is so big and so large and spreading so fast, then adaptive immunity is going to have to take over. So let's go over the key features to adaptive immunity. There are 4 key features to adaptive immunity. First one is adaptive immunity, if you remember from day 1, is specific. It allows it to focus its responses on a very specific pathogen that's actually present in the body. It is diverse and enables the body to respond to a variety of pathogens. It distinguishes self from non-self, so it prevents it from destroying self cells. And it has immunological memory. It allows it to respond more effectively if it happens again. If you get that same pathogen again, the body will remember and react faster. All right, so let's go into the specificity. The specificity is the reason why it's specific is because of 2 different lymphocytes, the B lymphocyte and the T lymphocyte. T cell receptors and the antibodies that are produced by B cells will recognize and bind to specific non-self substances. These non-self substances are called antigens. This interaction initiates a specific immune response. Each T cell and each antibody producing B cell is specific for a single antigen. So everything is very, very, very specific. So let's go into a little bit of what an antigen is. Antigens have sites that the immune system, or antibodies, these are antibodies, recognize. These sites that the immune system recognizes are called antigenic determinants, which is pictured there and the first arrow at the top, or epitopes is another name for it. Antigens are usually proteins or polysaccharides, okay? And a bacteria can have multiple antigens on them. Okay, so an antigenic determinant is a specific portion on the antigen, like a certain sequence of amino acids. All right, second type or key feature of the adaptive immunity is diversity. Adaptive immunity can attack all kinds of different pathogens. Different types of pathogens could be viruses, bacteria, protists, fungi, multicellular parasites, and any toxins that are made by these organisms. The human can respond specifically to 10 million different antigens from these pathogens. And what they do is they activate these lymphocytes. So humans need to generate a vast diversity of lymphocytes that are specific for different antigens. This diversity is generated primarily by DNA changes. Each B cell is able to produce only one kind of antibody. So therefore, there are millions and millions of different B cells in the body. There are also millions and millions of different T cells with specific T cell receptors in order to get all of these antigens. Antigen bonding will select a particular B or T cell for growth or spreading. Proliferation is called. For example, when an antigen fits the surface receptor on a B cell and binds to it, the B cell is activated. Then it will divide and form a clone of cells. This is called clonal selection, all of which produce and or secrete antibodies with the same specificity as the receptor. This concept also applies to T lymphocytes. This podcast will end. Please continue with podcast 2.